So thank you all for being here today and everybody who's joining on Zoom. I'm going to be telling you all about my research this summer in the Nelson Lab, which has focused on the characterization of precursor snow RNA transcripts with active translation in Arabidopsis thaliana. So as many of us know, plants have thousands of genes that contribute to plant form and function, and the end product of many of these genes are proteins. However, some genes produce RNAs that instead of producing a protein, serve some sort of direct biological function. A class of these are called link RNAs or long non-coding RNAs. And because of their low abundance and poor sequence conservation, as well as a lot of historical constraints in accurately annotating the genome, they've been less studied compared to protein coding genes. Some link RNAs contain short open reading frames that produce a functional SEP or SORF-encoded peptide that may be, we have evidence that they may be translated, but we're not sure which ones are or aren't, which makes these a huge focus in our research. So the annotations of the Arabidopsis genome are constantly being updated as we identify RNAs and other functional sequences that we've previously missed. In 2022, over 20,000 putative link RNAs were identified in Arabidopsis in a reannotation of the Brassicaceae genome that was actually done by members of the Nelson Lab in Palos et al. And these were not previously recorded. Many of these are only expressed in certain tissues or environmental conditions, such as drought or heat, which suggests that many link RNAs play specialized regulatory roles. And although link RNAs are generally defined as non-coding, some of the identified transcripts were found to have ribosome association and evidence of translation. In some cases, this suggests that they may not be true link RNAs, but they instead encode for small peptides from short open reading frames. And within this group of translated link RNAs, a smaller but biologically interesting subset were found to also function as precursors for small nucleolar RNAs or SNO RNAs. In the Nelson Lab, these transcripts are called PSATs, or precursor SNO RNA transcripts with active translation. So this is kind of more what that looks like. The larger teal transcript is what is actually being translated, and then these small pink sections are functional SNO RNAs. SNO RNAs are structural RNAs that are found in the nucleolus, where they direct the processing and chemical modification of pre-ribosomal RNAs. In Arabidopsis, there are two types of SNO RNAs, a CD box and an HACA box SNO RNA that are based off of their conserved sequence motifs, and they both function to do RNA modifications. So all of that together brings us into our big research question, which is trying to figure out exactly what these SNO RNA precursor transcripts are doing and what the translation event is. We're not really sure. But to investigate this, we focus on four kind of areas of experiments. We look at the expression of SNO RNA in a gene called pStat1 that I'll get into in a little bit. We also look at root germination and heat stress experiments to characterize pStats. And then lastly, we're also looking at breeding more homozygous mutants of pStats. And I'm going to get into all these points shortly, but we're going to start first with focusing on this first kind of experimental area, which is exploring this through pStat1. So pStat1 is a gene identified in the Nelson Lab through high throughput phenotyping of tDNA insertion lines that disrupted the candidate genes that were identified in Palos et al. And this revealed a precursor SNO RNA transcript with active translation, or pStat1, which is right here. When pStat1 is transcribed, it can be processed into these two SNO RNAs, again represented in the pink, theoretically a CD box and an HACA box. And then it also can be processed into a putative peptide, and it all undergoes active translation. So pStat1 mutants have severe developmental and reproductive phenotypes. They lag behind their wild type counterparts throughout early development, and they never quite reach the growth of the adult. On this picture on the left, this plant right here is a adult wild type, and this little one is the pStat plant. So it's extremely smaller. It looks drastically different than what we would expect. They also appear to have a defect in the formation of their cotyledons. And they do not produce pollen or siliques, which renders the plant infertile. Additionally, the heterozygotes can self-fertilize, but they have a subset of seeds that will self-abort during development. And we're not really sure what the cause is for these drastic phenotypes. But our hypothesis is that this has to do with those two SNO RNAs in the transcript. We think the functional SNO RNAs are dysregulated in the mutant. Transcription is occurring at the gene, but the transcript is not being processed to produce the SNO RNAs. So to test this hypothesis, we need to be able to verify that the SNO RNAs are being expressed in the mutant. And if they're not, this would be implicative that SNO RNAs are responsible for all of those reproductive and developmental phenotypes that we just saw. And in order to do this, we perform an experiment called a northern blot, which is very lengthy, but it involves separating RNA first using gel electrophoresis, and then transferring that to a membrane and then hybridizing it with probes that's going to detect our RNA. The reason we use a northern blot is because it actually allows us to separate SNO RNAs from the precursor transcript by their size. So as you can see here, this is our pSET1 gene. Theoretically, these two probes would bind to our two SNO RNAs, and they would also be binding to the precursor transcript as well. And then we'd be able to detect that using the northern blot. So this image right here is actually the final image of our northern blot. We set one up using wild type RNA instead of the pSET mutant because we wanted to be able to test our probes and see if they actually worked. So from what you can kind of see, there's nothing in these two lanes at all, which was just flower and leaf RNA from Arabidopsis, but there is this thick like band or blot for both of these probes in the positive control, which was PCR amplified pSET1. So what this tells us is that our probes work, but we need to tinker around with this experiment, the experimental design, to figure out an optimal RNA concentration. This kind of tells us that SNO RNAs are expressed maybe at a lower level than other types of RNA, and so we need to start with a higher concentration in order to see bands. So that's what we'll be doing with this in the future, and then hopefully when we're able to determine that optimal concentration, we'll be able to move on to looking at the actual pSET1. So besides pSET1, we have identified a ton of other pSET lines identified, again, in the Palis et al. That could also be crucial for plant development and stress responses. One way we wanted to determine that was by looking at root length and germination. And specifically, the reason that we chose that is because of kind of the data we see represented in this heat map. This shows the expression of pSATs across various tissues. And then I'm going to direct your attention to kind of this column right here that appears to be more expressed. And this is happening in germinating seeds. So that's kind of the basis of why we chose to look at that. So I wanted to explore that further. For that experiment, we used 8 different lines of pSATs from 4 different genes. So on this figure, these are the 4 genes. Again, this larger teal transcript is the precursor. These pink boxes are SNO RNAs. This blue rectangle shows where the short open reading frame would be. And then these green arrows are showing where the tDNA insertion sites are. And those end up actually being the names of our pSATs. So that's just a list of the ones that I used for these experiments. And so I recorded the root growth by taking scans at random time points over a 2-week interval. And then I made visual observations comparing the mutant lines to the wild type as a control. So I put up a few pictures here to look through super quick of the plates that turned out a little neater. And there's not really anything of note in differences between these lines and our wild type. So that kind of tells us that maybe pSATs don't really play a role in the root germination in these. But we would like to in the future perform some root tracing to see if there's any statistical differences and are maybe just not really visible upon looking at them. We'll be using software like ImageJ and SmartRoot for that, which would involve like taking the scans of the plates at the time points and tracing this main root and then collecting those numerical values for analysis. Besides the root length and germination assays, we're also looking at heat stress experiments. Our reason being is that there was a weighted gene co-expression network analysis that was performed previously, with various developmental and stress data sets, according to Palos et al. We decided to look at heat response because of the large number of p-stats that are associated with genes that have that expression. So similar to the root experiments, we use the same p-stat lines, and again, that's where they are on their genes. And then here is a round of these heat stress plates that were taken a week after the heat stress. So this process kind of involved a lot of trial and error to find like optimal conditions to use for the heat stress. But we ended up plating like the same number of seeds in each of these quadrants, and then each plate had a wild type that would act as the control. And we placed them in a 40-degree water bath for an hour, and then again, we observed this 1 week after. So what we're looking for with these is that the p-stat lines should maybe not look as great as the wild type post-stress, because that would tell us that there is something important that the p-stats are doing that is helping to regulate the plant after heat stress. So we're starting to see that trend emerge. We're looking at this first plate, the wild type seems to be growing maybe like more abundantly than compared to these two lines. We're also seeing this here. It looks like maybe the leaves are wider and they're kind of flourishing, and here it's maybe not looking so great. So this is a really great trend that we're going to look into more. We're hoping that we'll perform some more of these heat stress experiments, make more replicates in order to back up what we're seeing. So lastly, I have been observing new p-stats from seedling to adult to kind of see if there's any phenotypical changes happening there. And then with those plants, the reason that we have them is because I am selecting them for homozygous mutants so that we'll have more p-stat seeds to perform those phenotypic experiments, such as the heat stress and the root growth. So here is an example of one of the lines, which is Salk 025080. I would design primers for the mutant and wild type sequences, and then through DNA extraction and gel electrophoresis, look at where those bands are. And then if there's, say, a band in the mutant that we're not seeing here, that would kind of tell us that that's a mutant plant. And then we would keep those, grow them up and collect their seeds and use that in the future. So back to our main question being tested, we are attempting to determine what these SNO-RNA precursors and their translation event is doing through verifying SNO-RNA expression in pStat1, looking at root length and heat stress, and as well as breeding these new homozygous mutant lines. So in the future, we're going to continue tinkering with the experimental design of the northern blot to find the correct RNA concentration, again, so that we can actually check for those SNO-RNAs in the mutant. We found that maybe pStats don't really play a role with root length, but we're going to continue with root tracing to look for a statistical difference. And then again, we did find that trend that the wild types seem to be faring better after the stress than our mutants. So we're going to continue to make replicates to back that up, and then continue with our genotyping and breeding of homozygous mutant lines to use in the future and hopefully characterize these pStats. So with that, I would like to thank the Nelson Lab and everyone who's guided me, especially Dr. Andrew Nelson for letting me into the RU, and Dr. Katie Lehigh from Gustavus Adolphus College for presenting me with this opportunity. And I'd like to give a very big thank you to my research mentor, Alyssa, for all of her guidance and patience throughout this summer. And if you guys enjoyed my talk, to connect with me on LinkedIn. I love to see the research that's happening and grow my connections. So thank you all for your time. Love to take questions.
Based on your research, do you think that an increased expression of plants would possibly create a more heat-resistant plant?
I'm not really sure. I don't think like an increased expression would correlate to that. I think first we'd have to like have more replicates and be able to like confidently say like this is what happened, this is what is happening. And then I think that data and like us and adding those annotations would help any like functional researchers be able to like who are looking into like heat stress be able to use those for their experiments. Nicole?
That was absolutely fantastic. I have a question. So with your root germination experiments, Would you potentially consider looking at microscopy later on for information from secondary roots to see if there's anything different going on there, as well as the root tracing?
Yeah, I think that would be a great direction to go into. I think we also talked a little bit about kind of like the direction that like the tip of the root even grows, could have some sort of influence. I think that like if we were able to maybe not determine a sort of statistical difference with the main root, those would be good second options to look into. But also going back to that heat map picture. There's also, I think, like other tissues that could be looked at. We could do something maybe more focused on the seeds or just like other patterns of expression. Look at those tissues as well. There's like a lot of directions that we could go in, but I think that's a really great idea.
Thank you.
Abby?
When you were screening for your PSTAT mutants, were you interested in both your potential homozygous and heterozygous lines?
Yeah, so with the genotyping, I actually kept wild type and heterozygous plants as well, just as a control. We did see with PSTAT1, as I mentioned, it's not on here, but the heterozygous Cs are self-reporting. So it's possible the heterozygous phenotype is also affected in these. And then we kept wild type just kind of as a comparison. So yeah, we are definitely keeping those.
Amazing work and also amazing presentation. It's very cool and super easy to follow. I've never worked with Arabidopsis, so this might be a very dumb question, but in both your root test and your heat stress test, you had the seedlings in like in vitro conditions, right?
Mm-hmm.
Would you consider a mission, maybe for example, one of the ones that didn't do really well with the heat stress, to look at that in like older plants, maybe like outside of the plate, like just like
Like continuing to grow them after the stress?
Exactly.
That's really interesting. That could be a really great direction to go into. I think for my purposes here at the summer, it didn't really go beyond the in vitro. A lot of the mutant plants that we saw kind of had a hard time germinating on their own without the use of like the media and the plates. So I think like if they continue to grow and we can transfer them to soil and observe them that way, I think that would also be a really great direction to see like long term. But I think the purpose of like the quick stress with the water is to show us like short term effects because we're able to observe those changes so rapidly. But yeah, that would be a really great direction.
I have a question. How conservative is RNAs across species? Are you doing, what you're doing, does that matter for cabinets or tomato or corn?
Mhm.
They have the same sequences.
Yes, that's a really great question. The Palos et al. Paper that a lot of this is based off of did find that link RNAs are conserved across species. And then, so I think across Brassicaceae specifically, there was a PSAT we used called Link Cobra 1 that's not annotated on any of these. And that's because that was found by a different lab. And that's a PSAT that's across Brassicaceae. So they're definitely, I think, present in other, not just Arabidopsis, but in that family. Yeah. Chloe?
I do have a graph that showed why you were into stress. Were you guys also like
That's a great question. I think that would be an appropriate one to look into, considering there's also 7 PSATs that are associated with that. But again, like for my purposes this summer, it's not possible to attack everything. Was looking at hit response, but I'm sure that would be looked into in the future as well. Yeah.